Ooh, pretty good audience. Everyone wants to hear from Mike. I'm happy. Okie dokie. So I'm gonna start because we have enough people here. So as I said, good morning, everyone. It's great pleasure to be here one more time. So we are in the third day of the conference. Yesterday was like amazing. So many good people, so many great talks and great interactions. I'm so glad of being part of this society and of this conference. A little announcement before we start the session, because I know that towards the end of the session, I'm going to make these announcement again, but I know people usually leave because we have a tight schedule. So just for everyone, for especially for these students, make sure that you visit the exhibiting hall. We have videos in honor to pharmacologists, that people that create this society. So like for you young people that want to be inspired, go to the exhibiting hall, watch videos about Professor Maurício Rocha Silva e Sérgio Ferreira. So like two great, amazing Brazilian scientists that you should know. And also visit the young committee booth. So we have a stand in the visiting hall, Esbeft Jovem. We have votes going on for the second cultural contest for scientific dissemination. So it's important that you guys go there, visit the link, and vote for your favorite video because these guys made an incredible job. And also we have a raffle going on for a book. So you should, visit this stand and participate on it. Don't lose it. Okay, so without further delays, I'm gonna start the session, and I have the great honor of presenting the speaker today, which it is Michael Katerina. I'm gonna project to you guys this screen that has like a brief history of Mike. I'm so glad that I have the opportunity to call him Mike. And then today I decided to do like a different things. I'm gonna name all of the positions, all of the hats that Mike's wear because there are so many. He's an important person. All of the papers he published, all of the discoveries that he has made that have been so important for the scientific community. I decided to do differently. I will let the screen you guys can read all of these important things about him. I decided to say something cool about Mike because he's a cool person. So for us, at least for me, and then for most of the society that works with the pain and inflammation and then works in the field, probably know Mike. So Michael Katerina is like one of the guys that discovered the TRPV1 channels, right? So like you guys probably know him by reading him already. And then like for people that work with that, it's impossible not to have referenced Mike at least once in your life. So like I can give you a number of examples like 1997, Katerina 2003, 2006, 2007. So like those are like important references that I used for my PhD dissertation, and I'm pretty sure that you guys had like know already. So what I want to say here, like when I invite Mike to speak, is that as a brilliant scientist that he is, he's also a brilliant person. So I have the honor of working with him in the Department of Neurosurgery, which he is the boss. So like he's my big boss, and I'm so glad for it because he's an amazing person, and it's my greatest pleasure of introducing him today to give you this talk. Mike, the honor is yours. You're a mood, Mike. Sorry.
Thank you. It's a real privilege to be here today. Thank you so much for inviting me and for the overly kind introduction. It's been it's been my privilege to get to work with you and see your science really explode. And it's I'm fascinated to see where things go in the years ahead. I'll begin by saying bom dia, everyone. It is really wonderful to get to share with you some of the work that we've been doing in the lab over the last few years. I'm just going to share the screen here. Is that showing? Wonderful.
Looks
Thank
good.
you. So the main theme of research in my lab in over the last 6 or 7 years has really been in the field of trying to understand mechanisms of cutaneous pain. What are the mechanisms by which pain in the skin arises, and how does that pain undergo alterations following injury or inflammation to produce a state of chronic pain? And so I'm going to tell you about two broad areas today. The first has to do with the cellular molecular mechanisms underlying neuropathic pain. And this is just an oversimplified schematic to show some of the alterations that happen after peripheral nerve injury. So these represent two neurons that are innervating the skin. And when you have injury to one of these neurons, this sets in motion a number of different alterations that change the processing of painful stimuli and even non-painful stimuli by the peripheral nervous system. The injured neuron itself can develop it, can help contribute to the formation of a neuroma. It changes its intrinsic behavior. It can spontaneously fire. It will contribute to sensitization of the spinal cord dorsal horn so that you get central sensitization. It'll undergo wall area degeneration distal to the injury, and the target tissues become denervated, and that produces changes within the target tissues themselves. But also, these neurons don't exist in a vacuum. There are other neurons nearby that sense the damage to these neurons, and they themselves undergo changes. And it's the composite of all of these different events, both neuron intrinsic and neuron extrinsic events, that can lead to the alterations in the processing of exogenous stimuli that lead to neuropathic pain. And one of the layers of complexity here has to do with the large number of cell types that might be involved. So within sensory ganglia, which innervate the skin, these sensory ganglia are called dorsal root ganglia. For those of you who may not be in the field, the dorsal root ganglia lie adjacent to the spinal cord. And we have a pair of dorsal root ganglia at every level of the vertebral column. So that's where the cell bodies of these sensory neurons are located. And these neurons are actually quite heterogeneous. There are at least 10 different subtypes in the mouse, probably 11 or 12 in humans by latest count. Some of these neurons are specially tuned to detect noxious stimuli. These are what are called nociceptive neurons. Other neurons are predominantly involved in the sensation of non-painful temperature or light touch. The latter are called low threshold mechanoreceptors. And even these neurons aren't the only potential players here because they're surrounded by a number of other cell types. These include epithelial cells in the skin, like keratinocytes, specialized cells at the base of the epidermis called Merkel cells, that we'll talk about in a little bit, a whole host of immune cells that reside within the dermis and the epidermis, and other cells like glial cells, vascular cells, and fibroblasts. All of these cell types can communicate with one another, particularly following a nerve injury, and lead to changes in how information gets processed, and therefore lead to the phenomena of spontaneous pain and allodynia and hyperalgesia. So one cell type that we became interested in a number of years ago were Merkel cells. These are the cells, I'll just go back to this slide, that reside here at the base of the epidermis. They're derived from the keratinocyte lineage, and they're innervated by a subpopulation of low threshold mechanoreceptors called SA2 or SA1 or slowly adapting type 1 low threshold mechanoreceptors. These are large diameter neurons who, like the other sensory neurons, have their cell bodies in the dorsal root ganglia. And Merkel cells have been recognized for a long time to be involved in touch sensation. Female mice lacking Merkel cells show impaired texture discrimination. That's work from Steve Marriage's group. Merkel cells contribute to the sustained phase of touch evoked in SA1 neurons. So I mentioned that these Merkel cells are in a complex with the SA1 low threshold mechanoreceptors. And if you remove the Merkel cells or you incapacitate them, the SA1s can still respond to mechanical stimuli, but that response is altered. And finally, Merkel cells embryologically release BDNF to maintain the phenotype and the firing characteristics of these SA1 neurons. But what hadn't been clarified is whether Merkel cells play any role in chronic pain states, because we know that in the setting of nerve injury, even low threshold mechanoreceptors can contribute to mechanical hypersensitivity or allodynia. And so, as a start to approach this problem, we ended up taking a look, first of all, at what is the distribution of Merkel cells in the skin. And this is work that was carried out by Sungmin John, a junior faculty member of my lab. And what Sungmin did was to simply do immunostaining of mouse paw skin for Merkel cells. And I won't go into all the details here, but basically there are several different territories within mouse skin. We're looking at the lower surface, the plantar surface of the hind paw of the mouse. These orange structures are the foot pads. Behind those foot pads towards the heel is what's called the glabrous skin. In between the foot pads, there's an area that actually has hair follicles in the mouse. And so we refer to that as hairy plantar skin. And then wrapped around the top of the foot, and it's unraveled here, are these structures where there are hair follicles, and that's the hairy skin on the dorsum of the paw. And so what you can see here in the stain with keratin 8 antibody, which detects Merkel cells, there are Merkel cells scattered throughout the glabrous skin territory. And they're interspersed among nerve fibers, in this case, these myelinated nerve fibers. And if you look over on the right-hand side, what you see here is that those Merkel cells in green reside at the tips of these specialized terminal structures of tract C lineage sensory neurons. And these include those SA1 low threshold mechanoreceptors that I mentioned. And when Sungmin inventoried where these Merkel cells were, he basically found that they were in all 4 of these paw skin territories. Now, the model of neuropathic pain that we use in our lab, and that gets used in many labs, is a model that was developed by Isabella DeCostert and Clifford Wolf many years ago, called the spared nerve injury model. And this is a model in which the sciatic nerve of the mouse, this large nerve that innervates the hind limb, is injured, but it's injured in a very specific way. There are 3 branches to the sciatic nerve: the common peroneal, the tibial, and the sural nerves. And in this model, the common peroneal and the tibial nerves are transected, and a little piece of them is removed, but the sural nerve is left intact. And so what ends up happening then is that the middle of the paw gets denervated, but the lateral surface of the paw, which is innervated by this spared sural nerve, interestingly becomes hypersensitive. And so this is a beautiful model of long-lasting neuropathic pain. And what Sungmin was able to show is that when he performed this surgery and he looked at the middle of the paw where the tibial nerve used to innervate, you can see that by 1 week after surgery, there's a reduction in the density of nerve fibers in the middle of that paw. But interestingly enough, over time, the Merkel cells that were located in that territory disappear, and they're gone by about 2 months. However, in this lateral sural territory where the innervation is preserved, you can see here that the Merkel cells are preserved as well. So in order to address whether Merkel cells play any role whatsoever in neuropathic pain, we took advantage of a genetic model that was originally developed by Steve Marisic and his colleagues, in which the transcription factor ATO1 is knocked out within the epithelial lineage. And when you do that using a keratin 14 Cree driver, you prevent development of the Merkel cells. So here is glabrous skin from a wild type mouse on top, and then on the bottom, a Merkel cell knockout mouse. And one thing I want you to notice is that even though you've knocked out those Merkel cells, these sensory nerves appear to be relatively intact. So you can see these complicated ending structures of these SA1 neurons. And even though the Merkel cells aren't there, those neurons are there. And these nociceptive or pain-sensing neurons that express CGRP are also preserved under these circumstances. So what Sungmin did was to perform the spared nerve injury model in these mice lacking Merkel cells. And the results are shown here. This is a behavioral assay in which we're measuring mechanical sensitivity of the mouse paw. And we're doing this by poking the bottom of the paw with nylon filaments, with nylon filaments of different forces. And if you focus your attention on the black traces here, what we're showing here is what percent of the time does the mouse withdraw its paw at a given force. And so you can see that at weak forces, control animals barely respond at all, but at increasing forces, the prevalence of responding to that mechanical stimulation goes up to 100%. And these assays are actually being done in this lateral section of the paw where the innervation is preserved. And so, again, if we just follow the black columns here and we're looking at male mice, we see that after nerve injury, 1 month after nerve injury, this whole curve has moved to the left. This is a pharmacology conference, so I figured it was important to show a dose curve. And that's exactly what this is. So you can see that even at low forces, now the mouse responds about 80% of the time, whereas before injury, it only responded about 20% of the time. Interestingly, however, if we do the same analysis in mice that are lacking Merkel cells, there's no consistent significant difference in mechanical sensitivity at baseline. But after nerve injury, you see that there is less hypersensitivity, particularly at these low forces, in the Merkel cell knockout mice compared to wild type controls. And this is plotted on the right hand side over time. In this case, I'm just showing the data for this very weak force. And you can see that over time, the response in the wild type increases and plateaus by about 3 days and stays steady for a month. And the response in the mice lacking Merkel cells still increases, but to a lesser extent than in the control. So this tells us that at least in male mice, there is a necessity for Merkel cells in order to get normal mechanical hypersensitivity. However, in female mice, the story is different. You can see that in female mice, there are no differences between wild type mice and mice lacking Merkel cells. And so this suggests that there may be a sex difference in the relative importance of the Merkel cell SA1 neurite complex to mechanical allodynia after nerve injury. There's another thing, though, that we noticed in these experiments that really led us down a completely different path of investigation. And that is, as you see here, 1 week after nerve injury, you can see that the neurons have started to disappear in the middle of the paw. But over time, at 28 days and at 56 days, this space starts to fill in again. And so after denervation of skin, there are actually two ways in which the tissue can become reinnervated. The mechanism that's best studied is just classical regeneration. And what happens here is that the injured neurons begin to grow back along their original paths to reinnervate the tissue that they had innervated originally. So this is classical regeneration, and it's been the subject of study for many, many years. But there's a second process that is much less well studied, and that's called collateral sprouting. And collateral sprouting, as the name implies, involves the ectopic branching of these neighboring uninjured neurons. They recognize somehow the denervation of this tissue, and they send off new branches to reinnervate that tissue. And in the spared nerve injury model that we're using, regeneration is impossible because of the way that we injure the nerve. And therefore, any reinnervation that we see must be the consequence of collateral sprouting. And so this, again, is just a reminder of this model, just to show you that this territory in the middle of the paw that was originally innervated by the tibial nerve, after injury, loses its innervation. So there's a loss of sensation there. And the two areas of skin I'm going to talk about are this glabrous skin that's at the heel of the paw and this plantar hairy skin that's in between the foot pads. And so this is a picture of glabrous skin before and at various time points after spared nerve injury. And you can see, just as I showed you before, in this case, this is immunostaining for calcitonin gene-related peptide, which is a marker for peptidergic nociceptors, both C fiber and A delta fiber nociceptors. And you can see that there is a loss of innervation in the middle of the hind paw, but that by 1 month, it's starting to come back. And by 56 days, it's almost all the way back. The same thing is true if we immunostain the skin for a neurofilament heavy chain using an NF200 antibody. This labels both myelinated nociceptive neurons and low threshold mechanoreceptors. And we'll talk about the distinction between those later. But basically, you see the same phenomenon where there is complete denervation in the middle of the paw, but by 1 or 2 months, you see that that innervation is restored. The same is true in this patch of hairy skin in between the pads that's magnified here. You can see that there is disappearance of both CGRP fibers and neurofilament fibers after nerve injury, but it returns back towards normal by day 28, excuse me, and is nearly fully restored by 2 months. Now, how do we know for sure that this is due to collateral sprouting and not due to some sort of regeneration that we hadn't controlled for? Well, this is an experiment that Sungmin did to control for that. So what he did was he showed, first of all, 8 weeks after nerve injury surgery, you can see that in the spared nerve injury model, the tissue has become reinnervated. However, he took some of these animals and then did a second surgery in which he cut the sural nerve. And now you can see that if you cut the sural nerve, all of that renewed innervation of the tissue has disappeared, implying, or nearly all of it has disappeared, implying that the sural nerve is contributing to that reinnervation. The remaining innervation that you see here, we believe, is coming from the unrelated saphenous nerve, which is on the other side of the paw. So this genuinely is collateral sprouting. How far do these neurons go in restoring their original innervation patterns? Well, one thing that Sungmin has noticed, these are transverse sections through paw skin. One thing he's noticed is that if you look in the epidermis, at baseline, you have nice innervation you have neurons that penetrate between the keratinocytes and innervate the epidermis. Those neurons are gone after nerve injury, but you can see now by 8 weeks after nerve injury, we're starting to get some of that epidermal innervation back. Now, the neurons that I've been showing you so far are predominantly the CGRP positive neurons that most of which are nociceptive or pain sensing neurons. What about the low threshold mechanoreceptors? There are multiple populations of low threshold mechanoreceptors, and we have genetic tools, many of them developed by David Ginty's lab, who's now at Harvard, to visualize these LTMR subtypes. And Sungmin was able to show that in this hairy plantar skin, before injury, if you inject a retrograde tracer, fast blue, into the middle of the paw, and then give that dye about a week to get translocated back to the cell bodies of these neurons, you can label a variety of these different low threshold mechanoreceptor populations. So we have track C labeling neurons that have a couple of different subtypes involved, split Cre neurons, which predominantly consist of rapidly adapting A beta nociceptive neurons, heavily myelinated neurons, and track B lineage neurons, which consist of what are called A delta low threshold mechanoreceptive neurons, which are lightly myelinated. And so Sungmin took a look to see whether these low threshold mechanoreceptors are able to carry out collateral sprouting after nerve injury. And this is a little bit messy because there are cell types besides the neurons that are getting labeled in the skin. But I want to focus your attention mostly on these long filamentous structures. Those are the nerve fibers in these 2D projections of confocal imaging from the skin. And so you can see that in naive skin, we see all three of these populations, the track C population, the split Cre population, and the track B population. 1 week after injury, all of those nerve fibers disappear, and all that's left are these scattered non-neuronal cells that are cross-reacting with the tomato antibody. At 4 weeks after injury, you're still not seeing much. At 8 weeks after injury, you can see that in the case of the split Cree and the tract B Cree lines, there are still no filamentous nerve fibers that have innervated the skin, suggesting that these two populations of low threshold mechanoreceptors are completely unable to carry out collateral sprouting. On the other hand, in some animals, but not all animals, we see some degree of weak sprouting in this tract C lineage of neurons, suggesting that this is a particular population of LTMRs that may have some limited capacity to undergo sprouting. Another population of neurons that successfully undergoes collateral sprouting after injury are tyrosine hydroxylase positive nerve fibers. You can see again here, without going into too much detail, we can stain for them in the glabrous paw skin using either genetic methods or immunohistochemistry. They disappear after nerve injury, but by 2 months after nerve injury, we see reinnervation of that skin. Uh, there are multiple types of TH positive nerve fibers. Some of them are sensory neurons, and some of them are sympathetic neurons. But we believe that this reinnervation is predominantly by sympathetic neurons rather than sensory neurons because we can ablate them. Again, coming back to the theme of pharmacology, we can ablate them with systemic administration of 6-hydroxydopamine, which is a toxin that will kill off sympathetic neurons, but not tyrosine hydroxylase positive sensory neurons. Okay, and this is not an unprecedented finding. Work by Nascimento and colleagues back in 2015 revealed that in the rat following a spared nerve injury model, there is evidence for collateral sprouting of sympathetic fibers into the denervated skin. But it's transient in that case. It disappears by 8 weeks after SNI, whereas in our mouse model, that sympathetic sprouting appears to be persistent. So we know anatomically that these neurons make their way into this denervated skin territory, but are they functional? And the evidence for that is shown on this slide. This is a mechanical sensory behavior assay on the top left, in which Sungmin is stimulating the skin of the paw. In this case, he's focusing on that denervated patch of skin. Whoops, that denervated patch of skin in the middle of the paw, the hairy plantar skin. And he's stimulating it either with a high force filament or with a pin, with a metal pin. And you can see that the response to those stimuli initially is very robust, but after injury, within 3 days, the response pretty much disappears. But over time, over the ensuing month, you can see that response return back towards normal. Interestingly, the response never exceeds normal. So if you use a medium force von Frey filament to deliver a weak mechanical stimulus, you see that response goes away after injury. So in this case, the injured paw is in red and the control uninjured paw is in blue. And you can see that in the red traces here, the response goes away. It comes back, but it never exceeds the response in the uninjured paw. And we can see this in a different fashion here. This is looking at, again, the full dose curve of responses to mechanical stimuli over a range of forces. Prior to injury, there's a gray trace here that's kind of hidden behind the others. You can see this dose-dependent increase in mechanical responsiveness. 1 week after injury, the curve is flat. There's no mechanical responsiveness. But then 1 month and 2 months after injury, you can see that the response returns almost precisely to where it started. And then by comparison, just to harken back to some behavior that I showed you at the beginning of the talk, if you look right next door in this area of tissue where the neurons were spared, you see the expected enhanced mechanical sensitivity. So there's a difference between the mechanical sensitivity of this patch of skin, where the neurons have always resided, and this patch of skin, where the neurons have repopulated, despite the fact that it appears to be the same neurons that are innervating both pieces of tissue. So we think that's a fascinating issue. Finally, we can start to apply other technologies to address the functionality of these neurons. We can overexpress the light activated ion channel called channel rhodopsin in the sensory neurons. You can see the nerve terminals in the skin here, and we're staining for that channel-rhodopsin protein, and you see that it's gone by 1 week and it's restored by 2 months. And if we shine a blue laser light on the bottom of the paw, and these were experiments that we did actually with the help of Bika's current mentor, Alban Le Tremolier. And what we see here is that if we photoactivate these nerve terminals, we have a response before injury, the response disappears, but then it comes back over time. And so we think we have some nice tools now by expressing this channel rhodopsin in different subpopulations of neurons. We'll be able to start to deduce what the functional contribution of each of those neurons is to the mouse behavior. So just to conclude this first part of my talk, um, Merkel cells are found throughout the 4 territories of the mouse hindpaw skin. Denervation of glabrous skin results in loss of Merkel cells. But interestingly, in the spared territory, genetic absence of Merkel cells does not lead to loss of associated Traxia lineage nerve terminals, but it does result in reduced neuropathic pain in the spared nerve injury model. But that deficit is unique to male mice. Following spared nerve injury, collateral sprouting of spared nerves leads to tibial territory skin reinnervation, but it's not uniform reinnervation. Nociceptors and tyrosine hydroxylase positive sympathetic neurons sprout efficiently, whereas low threshold mechanoreceptors show little or no sprouting. And then finally, mechanical sensitivity is grossly restored within a couple of months to this territory, despite the fact that you have a very different composition of nerve endings in that skin and without a hyperalgesic overshoot during this period. Now, great minds think alike, and so we're not the only people who have pursued this question. And very recently, a group led by Rohini Kuhner in Germany published a paper describing very similar findings. They also observed that nociceptive neurons sprouted after injury, and non-nociceptive low-threshold mechanoreceptors failed to do so. Interestingly, they extended their study to much longer time points than ours. And what they observed when they plotted the threshold for mechanically evoked behavioral responses in those animals is that just like we saw up to about 8 weeks, there is a restoration of mechanical responsiveness. But if they waited for much longer times, what they observed is that eventually there's an overshoot such that there is an increased mechanical hypersensitivity or a mechanical pain phenotype in this tissue. And so this is something we're exploring currently in our experiments to see if we see the same thing. And although the studies largely show some of the same phenomena, there are differences. We're looking at different types of skin. We see sympathetic neurons sprouting in our experiments, and they haven't. We also observe this population of Traxia lineage neurons that sprout. And I showed you earlier that we observe penetration of the epidermis by some of these collaterally sprouting neurons, and there was no epidermal reinnervation in that study. So we're going to have to look more carefully to see what might account for the differences between these studies. But for the rest of the talk, I'd just like to change gears a little bit and talk about another broad topic in the lab. We've tried to take advantage of disease models in order to understand some of the more complex phenomena that are associated with pathological pain in the skin. And there's a family of hereditary diseases in humans called palmoplantar keratodermas, which basically means there's a thickening of the skin on the palms of the hands and the soles of the feet. And if you look under the microscope, what you see is that is a reflection of an expansion of the epidermis, the outermost layer of the skin. So this is healthy skin on the left, stained with hematoxylin and eosin, and you can see this relatively thin epidermis with the dermis down below. But in affected skin from this patient with palmoplantar keratoderma, you see this massive expansion both of the viable keratinocytes, the live keratinocytes, and the dead cells, the stratum corneum that's superficial. So palmoplantar keratodermas, or PPK, can be caused by a variety of things. There are hereditary PPKs. There are at least 25 different genes in humans that can lead to this condition. And then there are also acquired forms of PPK. Certain drugs or certain forms of cancer can lead to thickening of the skin on the hands and the feet. But we were intrigued by the fact that among the hereditary PPKs, there is quite a degree of heterogeneity in whether or not these lesions are painful. Anyone who's ever had a callus on their skin knows that very often, if you have, like I have a writer's callus on my finger, it's less sensitive than my other skin territory. When we think about calluses as being an adaptive response to repetitive trauma that deadens the sensation in that tissue. But interestingly, in some cases of palmoplantar keratoderma, there's exquisite pain associated with these lesions. So, two of these conditions that have very frequent pain, one is called pachyonychia congenita, and one is called Olmsted syndrome. Pachyonychia congenita is caused by mutations in one of these three keratin proteins. Olmsted syndrome is caused by a mutation in one of my old friends, the ion channel TRPV3, or by membrane protease. And then there are other forms of PPK that where pain is seen only in some patients. So mal de Maleta is due to mutations in this protein called SLRP1. Epidermolytic PPK is due to mutations in keratin 9. And pain is present in some of these patients, but it's not as prevalent as it is here. So we decided we wanted to try to understand this better. And I won't show you, we've done some studies together with Michael Polydefkis in the neurology department here at Hopkins to look at the neuroanatomy of these lesions in skin from patients with pachyonychia congenita. But what I'm going to talk about today is our work on mouse models of PPK. We have mouse models that genocopy some of these conditions. And I'll show you some of our preliminary data trying to characterize whether or not they have pain phenotypes. So these are some of the mouse models that we're working with. We have a keratin 16 knockout mouse line that is a genetic model to some extent of pachyonychia congenita. We have generated a mutant TRPV3 transgenic line in an attempt to model Olmsted syndrome. We have obtained SLRP1 and SLRP2 knockout mice, and I'll tell you more about what these are in a moment, to model mal de Maleta, and we have keratin 9 knockout mice to model epidermolytic PPK. Okay, so to date, most of our data have really focused on these mouse lines, SLRP1 knockout and SLRP2 knockout. So what are SLRP1 and SLRP2? Well, they're small secreted proteins. They're members of the Ly6 PAR family of secreted molecules. They're expressed in many tissues throughout the body. And although their physiological function is not thoroughly understood, they're thought to act as allosteric modulators of nicotinic acetylcholine receptors. And because nicotinic receptors are involved in epidermal homeostasis and differentiation, the absence of either SLRP1 or SLRP2 leads to that dyshomeostasis that results in palmoplantar keratoderma. And Steve Young's group at UCLA had generated two different mouse knockout lines, a SLRP1 knockout line and a SLRP2 knockout line. And what they observed was that these animals indeed show palmoplantar keratoderma. They show thickening of the skin that you can appreciate either in this macroscopic picture here of the mouse hind paw, or under the microscope, you can see that the thickening of the epidermis looks very much like what we saw for the human PPKs. And these two knockout lines really are indistinguishable from one another. They exhibit the exact same phenotype as far as we can tell. And the Young lab has gone so far as to make double knockouts, and there's no change in the phenotype beyond what you see in the single knockouts. But what hadn't been addressed in these lines was whether or not there are any alterations in pain sensitivity. And so this was a project that was undertaken by Rachel Weinberg, a former graduate student in my lab, who's now just started a job at the National Institutes of Health. And what Rachel showed using a variety of pain behavioral assays, including this one that I showed you before, the von Frey filament assay, Rachel showed that there is enhanced mechanical sensitivity in SLRP1 knockout mice and in SLRP2 knockout mice. So you see here, the black bars here represent the response frequencies of the wild type mice to different forces, and the blue bars represent the response frequencies of the mutant mice. And you can see that at any given force, there's increased behavioral withdrawal in the knockout mice compared to the wild type mice. They also show increased heat evoked pain behavior. So in this assay, we put the mouse on a glass surface and we shine a heat lamp on the bottom of its foot. And you see that it takes less time for the mutant mice to withdraw their paw than it does the wild type controls. And this is true in both the SLRP2 knockouts and the SLRP1 knockouts. And I've color coded here male and female mice, and you can see that the difference is true in both males and females. However, if we test heat pain sensitivity in another piece of tissue, in the tail, which, as far as we can tell, it does not exhibit a visible palmoplantar keratoderma, we do not see any difference in heat pain sensitivity in the tail of these mice. So this enhanced pain sensitivity is not a general consequence of SLRP knockouts, but rather it is a consequence of the pathology that happens within that paw skin. All of the pain behaviors that I've shown you so far are behaviors that are evoked by either poking at the paw or heating the paw, or we can also do it by injecting irritants such as capsaicin. But we also observed that these mice show spontaneous behaviors, suggesting that there may be ongoing or spontaneous pain even when they're not stimulated. So these are this is a distillation of some of those findings, but you can see that in both the SLRP1 and the SLRP2 knockout mice, there are more instances of flinching of the paws. If you just have the mice sitting quietly on a surface and you videotape them, they spend more time licking their front paws and they spend more time licking their hind paws. So we think that there is a spontaneous component to this pain. So, what are some of the mechanisms that might be underlying this? Well, we're just beginning to undertake this, but one of the things that we see is that there's a dramatic alteration in the immune cell composition in the skin, in the PPK affected skin of these animals. And I won't go into the details here, but basically a variety of different immune cell types are altered in abundance. So T cells, mast cells, and macrophages are elevated, and Langerhans cells, which are in the epidermis of the skin, are actually decreased in these animals. And so one of the efforts in the lab is to go back and individually deplete these different immune cell populations to see if that will affect the pain phenotype. We also see, though, using neurophysiology, that these neurons are the neurons from these animals are intrinsically hyperexcitable. And this is work done by Lin Tao Chu, our close collaborator who works right next door to us. Lin Tao has developed a preparation where he has an anesthetized mouse and he can visualize the sensory ganglia of these mice while he's stimulating the hind paw. And so this is just an image of the mouse in which these neurons are actually genetically labeled, or they're labeled with a retrograde dye. And one can either use optogenetics and study the calcium responses in these neurons, or in this case, what Lin Tao is doing is using a patch pipette and electrophysiologically recording from these neurons. And what he sees are two interesting things. First of all, even without any stimulation of the paw, there's a certain population of neurons in these mice that are showing spontaneous firing behavior. And that's interesting because, as I mentioned earlier, the awake mice show this spontaneous paw licking behavior. And so we think this might be a neurophysiological underpinning of that phenomenon. In addition, if you just focus on the neurons that do not show spontaneous firing, and you stimulate the paw skin in the receptive fields of those neurons with a mechanical stimulus, you see that there's an increased response to mechanical force over a range of forces in the knockout neurons compared to wild type. So not only do these mice show behavioral signs of pain hypersensitivity, but they also show the neurophysiological signs of that. Okay. So what's going on at a molecular level? Well, this is really where we're spending the majority of our time right now. And I'm going to give you the warning that I'm not going to have any positive answers for you here. I'm going to show you an example of a negative result. But basically, if we perform RNA-seq on the ganglia that innervate the affected hindpaws in these animals, there are about 40 genes that are either upregulated or downregulated in those sensory ganglia. And we've been able to confirm with a number of these genes that these changes are confined to the ganglia that innervate the affected skin. We don't see such gene expression changes in many cases in the ganglia that innervates, say, the trunk skin or other skin that's not affected by PPK. So this really is a PPK-driven process. The other thing I'll point out is that there are about 5,000 genes that are dysregulated in the skin of these animals. So this is a little bit of a more daunting task in order to study this and to understand which might be related to pain. And so we're focusing most of our attention here on the skin that is on the sensory neurons that innervate that skin. And one gene that, again, is another familiar gene to us is another TRP channel called TRPA1. We see that TRPA1 mRNA expression is elevated in the ganglia that are innervated, the affected tissue in the SLRP1 knockout animals. Interestingly, it's not altered in the affected skin of another PPK mouse line, the keratin 9 knockout animal. So we think there's something specific to this. It's also not altered in the thoracic ganglia, which innervate the unaffected skin. And that data that I just showed you, this is qPCR on total DRG. We originally observed an upregulation of TRPA1 in this RNA-seq data set, but we wanted to apply a third method, and that's RNA-scope in situ hybridization, to visualize which cells within the ganglion are showing this TRPA1 upregulation, because there are actually many different cell types in the dorsal ganglia besides just neurons. But indeed, it looks like the elevation is in neurons in the SLRP knockout tissue. You can see that quantified here. So does TRPA1 play a role in the augmented pain sensitivity of the SLRP knockouts? Well, so far, we don't think it does. And so here are just some distilled data. We're looking at mechanical sensitivity on the left and heat sensitivity on the right. And just to orient you, the mice that are SLRP knockouts are in blue, the wild type mice are in black, and the double knockouts, SLRP knockouts, TRPA1 double knockouts are in red. And what we see here is that knocking out TRPA1 does not rescue the pain phenotype of these SLRP knockouts, either in the mechanical assay or in the heat assay. If anything, there might even be a little bit more heat hypersensitivity in the double knockouts and the single knockouts. So the number of animals is quite small here. So this has to be confirmed yet. The other direction that we're going with this work is to take advantage of what I mentioned earlier, and that is that we have other mouse lines that are genocopies of human hereditary PPKs. So here are two of them, the keratin 16 knockout mouse line and the keratin 9 knockout mouse line. We obtained these from Pierre Coulombe at the University of Michigan and the keratin 9 knockouts from Erwin McLean at the University of Dundee. And what you can see is they have demonstrated already that there is PPK in the pad skin of the paws of these two animals. Now, in the keratin 9, the PPK is a little bit more subtle than it is in the K16s or the SLRP knockouts. The other thing that makes this a little bit complicated is that whereas in the SLRP knockouts, this overt PPK phenotype is seen both in the front paws and the hind paws, in the K16 mice and the K9 mice, it's really front paw predominant. And that's unfortunate because most of our pain behavioral assays really are focused on the hind paw. It's a little bit trickier to do pain behavioral assays on the front paw. But we have found one way to do it, and that is to use a laser stimulus to heat the front paw skin. And here you see that in the SLRP knockout mice, just as I showed you before, there is heat hypersensitivity. In the keratin 16 knockout mice, there is also heat hypersensitivity in the front paws. But in the keratin 9 knockout mice, as far as we can tell, there's no such hypersensitivity. Similarly, if we look at spontaneous pain behavior, there is increased spontaneous pain behavior in the keratin 16 knockout mice, but not in the keratin 9 knockout mice. So this gives us a nice opportunity because we can start to now cross compare these different mutant lines. They all show this PPK phenotype, but they exhibit uh, some shared pain behaviors and some different. So we can start to do comparative transcriptomics now on these lines. And this is work that we're doing in collaboration with Pierre Coulombe at the University of Michigan to see which altered genes are shared between the different PPK knockout lines and which are unique to one line or another. And we hope that by doing this, we can start to identify which genes are uniquely associated with the sensation of pain, of pain hypersensitivity, and which are not. And then finally, I just want to very briefly mention another model that we've been trying to make related to this disease called Olmsted syndrome. And this is a disease that is caused by mutations, gain of function mutations in the ion channel TRPV3. And it was originally identified as a contributor to a hair loss phenotype in a spontaneously occurring mutant in mice and rats. But then in humans, it was discovered to be associated with this really horrific disfiguring PPK. And the model that had been studied in mice before was a model in which there was this particular mutant, a glycine 573 to serine. But more recently, there was another model that was, or another PPK associated mutation that was discovered by Alain Hovnanian and his colleagues in France, in which there was a mutation at this leucine 673 phenylalanine. And we became interested in that mutation because not only do those individuals exhibit PPK, but even before they exhibit PPK, you notice they exhibit this intense reddening of the skin. And it's extremely painful. It's a phenomenon referred to as erythromelalgia. And so we developed a mouse model in which the TRPV3 mutant that had been identified by the Hovnanian group would be expressed under the control of its own promoter, but in an inducible way. So depending on what Cree driver line we use, we can trigger the production of this mutation in one cell type or another. And so for the first time, we're going to be able to address the cell type dependence of some of these phenomena. And I won't go through the genetics here, but I'll show you that what we observe in these animals, interestingly enough, is no development of PPK, despite the expression of this mutant TRPV3 in the keratinocytes. You can see that the epidermis is a unaltered thickness, but we do see this loss of hair phenotype. And when we look at the paw sensitivity of these animals, we do not see any mechanical hypersensitivity. So we do think that this is able to reproduce some of the phenomena associated with Olmsted syndrome, but we're not seeing a pain phenotype. And so now we're going back and introducing this mutant into the sensory neurons to see if that drives any alterations in pain in these animals. And so to summarize this last part of the talk, SLRP1 and SLRP2 knockout mouse models of multimodal exhibit a multimodal pain hypersensitivity. And we see a variety of alterations both in the immune cells and in the neurophysiology and gene expression in these animals. TRPA1 mRNA is elevated in the dorsal root ganglia of these mice, but does not appear to be essential for the pain phenotype, at least at the resolution we've tested. Keratin 16 knockout mouse models of Pachyonychia congenita also exhibit pain hypersensitivity, but shows patterns of changes in the skin transcriptome that are distinct and incompletely overlapping from those of the SLRP1 knockouts. Keratin 9 knockout mice, a model of epidermolytic PPK, shows no apparent pain phenotype, so we think these will be a great negative control. Inducible expression of Olmsted syndrome TRPV3 mutant in the epidermis gives us alopecia, but no PPK or pain phenotype. And so, taken all together, we're starting to accumulate a panel of mouse lines that we think are going to provide a nice opportunity for comparative mechanistic analysis of pain in hereditary PPKs. So with that, I'll just close by acknowledging the people involved in this work. The work on the Merkel cells and neuropathic pain was spearheaded by Sungmin John and Dennis Chang, who's a graduate student in the lab. The work on collateral sprouting was also done predominantly by Sungmin John, but with a lot of help by a number of colleagues, both at Hopkins, at the University of Wisconsin, including Alban Latremouille at Hopkins and Latasha Crawford at the University of Wisconsin. Work on pain and PPKs is spearheaded by Rachel Weinberg, but also involves Lin Tao Chu at Hopkins and Michael Polydekis and his colleague Baohan Pan. And these are the folks who paid the bills for the work, and work in the lab is also supported by the Neurosurgery Pain Research Institute in the Department of Neurosurgery. So with that, I will close and I will thank you very much for your attention.
Thank you so very much, Mike. Very elegant work. Beautiful, beautiful. I have no words to say. I've seen like parts of this presentation here and there, and then now that you told us this story, like the complete story, yeah, it's so beautiful. It makes so much sense, and I'm pretty sure that everyone in this room is delighted by the data that you shared with us. I'm very happy to say that we have like over 50 people in the room, like in terms of this is like a lot to me. So I'm very glad that people are like engaging in participating. To the audience, please, like ask your questions at the Q&A session that we have. We are, I'm pretty sure that Mike will be delighted of hearing your questions and answering them. So far we have one question already in the Q&A, and then I'm going to read that to you, Mike. And I also have a question for you because it's like keen to my heart, like the topic that you just brought to us. But let's start with the question from Amanda Siena dos Santos. So she asked you, Mike, regarding the part one of your talk, so like the first part of your talk, is there a physical limit for the reinnervation of the tissue? And then she says, for example, the lesion should be at 30 centimeters maximum, of the tissue for the neuron to be able to reinnervate. So she wants to know, like, if there is like a physical limit.
It's a great question, and the answer is probably yes. So in human skin, where the distances are much larger, there is evidence for sensory reinnervation of denervated skin by collateral sprouting. The literature has been a little bit mixed on this, but I would say that in some cases that degree of reinnervation has been limited in distance. But I don't have a good sense of whether that is an absolute situation. In other words, it's not clear that that's a generalizable phenomenon, or if it was only observed in some studies or in some pieces of tissue. I will say that one of our motivations for understanding this phenomenon is that if we can understand how to either promote the collateral sprouting of some population of neurons, or perhaps discourage the collateral sprouting of some population of neurons, we might be able to change both the area of skin that can be reinnervated by sprouting, and also we might be able to avoid some unwanted consequences such as long-term neuropathic pain.
Very cool. Thank you so much for your answer, Mike. So since we don't have any more questions in the chat, I'm gonna take this moment to ask you a question. As I said, into my heart, because I worked with TRPA1, and then this is one of the reasons that I was so motivated reading your work. And I was very intrigued by the data that you showed with the double knockout, like SLRP1 and TRPA1 knockout mice, and they did not show any behavior profile. So like ablating TRPA1 genetically did not do anything that you were expecting to these SLRP1 mice. So my question is like towards the pharmacology point, because I'm a pharmacologist and this is a pharmacology meeting. And then it is also like from my experience, because I work with both like the antagonists for the receptor and the the TRPA1 knockout mice. And then what I saw with, at least with my model, is that the knockout mice did not show any behavior phenotype, but then like when we used the antagonist, we saw some phenotype, and then we believe that knockout mice, there was some sort of compensation that was like preventing us from seeing it. So like my question is like, do you guys thought of using like antagonists so we could prevent some of this behavior?
Yeah, I think that's a great point and a great question. We and to answer your question, we've thought of it. We haven't done it yet because we've to some extent, Trip A1 is low hanging fruit. It has a well established role in pain, and so we've gone after some of the shinier objects that genes that might not be as well established as contributors to pain, and we got a little discouraged by the double knockout data, because then if we do see a distinction between the antagonist effect and the double knockout, we're going to need to explain that discrepancy, and we can explain it in
Exactly what I had to do.
exactly. So we're trying to pick our battles here, but I thoroughly agree. I think that there are other examples of where elimination of a gene or of a cell type fails to show a phenotype when acute antagonism doesn't. And another example of that is a molecule I'm more familiar with, and that's TRPV1. Knocking out TRPV1 doesn't do much to thermoregulation in animals. You have to look really hard to see little differences in thermoregulation. But if you acutely administer a TRPV1 antagonist, you get a transient hyperthermia, depending on the nature of the antagonist.
Yes.
So I think that that your point is an excellent one. That we very well might observe an effect of TRPV1, or contribution of TRPV1 to this pain phenotype if we were to take a pharmacological route. And the good news is there are good TRPA1 antagonists to try. So we need to.
Yes.
What I need you to do is at the next lab meeting, point to someone who has spare time around the table so that we can get them to do this experiment.
I'll do it. I'll be the
That
bad
sounds
guy.
wonderful. I'd love it.
Thank you so much, Mike, for your participation with us. It was truly an honor. I think everyone in this room is very delighted. I'll have to end the session because we're a little bit over time. It's like not a lot, so I'm very happy with. And then I thank you all for your attention. I thank you all for being here with us today. And then, like, again, last but not least, don't forget to visit the exhibit hall and, watch the videos that we have there. Go to the Young Committee stand, participated on the raffle for the book that we have, and vote for the videos that are there on our contest this year. So thank you so very much. Thank you, Mike, again.
Thank you.
I'll see you.
It's been a privilege.
Bye-bye.